So Safer Medicines Trust is a patient safety charity who aim to make medicines safer, and that the way we want to do that is by encouraging a change in the way that medicines are currently tested, because the current system is based on animal tests, which are not very predictive for humans, and we want to make the whole system based on human relevant science. And so there is our website with a huge amount of information on our website. I want to emphasise that we are an organisation of scientists and doctors, and we are very pro-medicine. That means that we recognise that many medicines are essential and save many lives, but we do also recognise that they also cause a lot of injury and even death from adverse drug reactions, sometimes called side effects, even when medicines are properly prescribed and properly taken by the patient. This is not talking about overdoses or misprescribing or any of those things. This is properly prescribed prescription medicines kill hundreds of thousands of people every year, in addition to all the good that they do for many people, but for other people they do an enormous amount of harm. And this is why I founded Safer Medicines Trust, not to challenge the idea that you will all be very familiar with, and you've probably seen this report a few years ago called Normalising the Unthinkable by the Oxford Centre for Animal Ethics. We don't challenge the fact that animal experimentation is one of the major moral issues of our time, but we want to help people to recognise that it's also one of the major scientific issues of our time, because this is really not recognised very much at all by politicians or by the public, or even by scientists, by many scientists a lot of the time. Okay. And so what does this have to do with animals? It has a lot to do with animals, because animals are still used in many ways in medicine. They're used for studying disease. They've been used as the whole basis of human medicine for decades, for studying diseases, developing treatments, and for testing them for safety. So this slide is asking, how are drugs tested for safety? And there are several different steps, and usually it starts with testing on the computer, which we usually refer to as in silico testing. And this covers a whole range of things. There are many different ways to use computers to model the effects of drugs, starting with the most basic thing, which is modelling the structure of the drug. And so you'll see the molecular structure on the screen, and this can be done to work out structure, activity, relationships, because what the drug does depends very much on its structure. And you can see that on the screen and experiment with it to see the effects that it might cause, even before you've ever synthesized the compound. So this makes a lot of sense, saves a lot of money. You don't even have to go into the lab and create the compound until you've synthesized it on the computer, played with it on the computer, looked at all sorts of things that it may or may not do with regard to whether it's effective, whether it's safe. Computer modelling becomes very much more sophisticated than that, so that you can have virtual patients on the computer and give the drug to a virtual patient in different concentrations and see what the effects might be. You can even run virtual clinical trials on hundreds of people, virtual people, on the computer. So it's an incredible technology, and it's improving all the time. Another major thing that is done, kind of the next stage or simultaneous stage, is to test, once you've got a new medicine, once you've synthesized a compound, well, it's not a medicine yet, it's a potential medicine, a candidate. You can test it in vitro, which literally means in glass, in the test tube, on cells, and preferably on human cells and human tissues. But a lot of this, of course, is done on animal cells and animal tissues, which can be a big problem because you have to then extrapolate from the animal to the human, and extrapolation does not always work. But the next stage, which should always happen, it should be mandatory, but it isn't yet. That's what we're working towards. But the next stage is to do more sophisticated tests. So the early cell tests can be two-dimensional. So it can be things like cells in a petri dish, flat layers of cells. And we now know that they are not very predictive because they don't represent what it's like for the tissue to be in the body. And so better, more sophisticated, more expensive models are now available, and they're all 3D, three-dimensional. Things like organoids and mini organs and organs on chips, all those sorts of things. These are very sophisticated tests, and we advocate that they should be used before moving on to the final stages of testing a new medicine for safety. And the current next stage before we move into people, and it's mandatory, it's a legal requirement, you cannot test a drug in people until you have tested it in animals first. And the idea is that we're protecting patients, we're protecting the volunteers in the clinical trials by testing on animals, and on a range of animals, rodents, so rats and mice, not always both, one or the other, and then a higher mammal, dogs or monkeys are usually used. And the idea of having a variety of species is that if it looks safe in all of those species, then it should be safe for humans. And on that basis, a drug company will be given authorization, a license, to move on to human clinical trials where the new medicine will be tested in humans, in volunteers and in patients. 12 years ago now, there was a landmark report from the US National Academy of Sciences called Toxicity Testing in the 21st Century. And they said the vision for the toxicity testing in the 21st century, articulated here, represents a paradigm shift from the use of experimental animals toward the use of more efficient in vitro tests and computational techniques. And needless to say, this paradigm shift has not yet happened, but this is what they said as well, which was very very prophetic. They said that policies designed to overcome tendencies to resist novel approaches and maintain the status quo will be important. And this is what I'm going to be talking about for most of the talk. And I'm just going to move the little picture of you up because it's kind of... oh, silly thing. It's... okay. There we are. So many things are preventing the changes that we need, and I don't know which is more important than any other. They're all very important obstacles, barriers to change. And the most obvious one is that it's always very hard to change traditions. People are... it's human nature. People are naturally very resistant to change, and they're comfortable with the status quo. It's what they're used to, and they naturally want to cling to the status quo because it takes less effort than learning a new way and changing habits, changing things that you're used to. There is a phenomenon called lock-in, and there's a wonderful paper that I think you've already looked at by Joshua Frank, which explains how technologies get locked in even when they're not the best technology. But there are many, many reasons why people get stuck in a rut on an inferior technology. And this is because institutions are built around it, and people's behaviour and psychology are locked into it because that's what they're used to. If you've had a biomedical education, most of your professors will have taught you that everything we know in biology we've learnt from animals. That's the way that they did it, that's the way they were taught, and they will naturally pass it on to their students. And things get passed on forever and ever and ever until somebody makes a major change, changes the paradigm. And where it says financial, that means, for instance, in academia, people giving grants, it's very difficult to get grants for research if it doesn't involve an animal model. We hear this all the time when people do research in human systems, and then they want to publish it in a journal, or they want a grant for doing some more work, and the journals and the funders always say, where's the animal stuff? And so it's very difficult to break through. And of course there are vested interests as well. It is a massive industry that breeds mice and other animals and sells all the cages and the equipment. It is a huge industry that employs hundreds of thousands of people, and naturally they don't want to all lose their jobs, and so they are very protective. It's a big industry looking to keep itself alive. And for pharmaceutical companies, they don't want their drugs to hurt people, but it is a massive problem. Adverse drug reactions do injure and kill hundreds of thousands of people, as we've already discussed. And so pharmaceutical companies often get taken to court by victims or by relatives of the victims. And the best protection that pharmaceutical companies have in court is to stand up and say, we tested this drug as well as we possibly could. We tested it in mice, rats, guinea pigs, cats, dogs, monkeys. What more could we possibly have done? And juries are persuaded by that, and so the pharmaceutical companies find a lot less money than they would be if the jury understood that all of that animal testing wasn't helping, and that actually the pharmaceutical companies should have been doing human-based testing. And then the final very perverse obstacle to change is validation. And validation is very important and necessary to make sure that any method that is used is robust and reliable and predictive and accurate. We're not suggesting that validation must be stopped, but it is a bottleneck, because it's very, very rigorous for new technologies. The big disconnect is that animal tests have never been validated, and yet new technologies, in vitro technologies, have to be validated by big studies that take up to 10 years and up to $1 million to conduct. And so it slows everything down. If you're a small biotechnology company and you've invented a good new technology, you can't afford to have it validated, and you also can't afford to wait all of those years while it is validated. And the trouble is that the field is moving so fast that by the time you've finished a 10-year validation study, that technology is already out of date. It's been superseded by newer technologies. So we definitely need to streamline validation, speed it up, make it much faster, and also get all the different industries and all the different countries to talk to each other, because they all have different requirements. And so validation that might suit the pharmaceutical industry might not suit the pesticide industry or agrochemicals, and or different countries. And so the same test will be validated over and over again in different countries and different industries. And the most stupid thing of all about validation in the past, anyway, was that the new method that is being validated is compared against what has always been seen as the gold standard, which is animal tests. And so if the...
Hey, sorry, you hear me?
Yes?
We just had a breakdown of the connection. Can you repeat the last sentence you were talking, please?
Okay, yeah. I was saying that the most stupid thing about validation is that new methods are compared to the gold standard animal tests, and that means that if the new test is better, it won't be validated, because what they're looking for is repeating the performance of the animal test. So very often the new tests are better, and that means they fail the validation. And so at last this is being recognised, and people are understanding that a new approach to validation is needed. And most importantly of all, the US regulator, the FDA, has understood that, and we're not alone in calling for a new approach. They're calling for a new approach now, and they're working on a thing called qualification, which is not as slow and expensive as formal validation. It streamlines it, and so this is much better. Are we okay? You can still hear me, okay?
Yes.
Lovely. And so they are recognising, even governments are now recognising that the gold standard has to be human biology, not animal biology. And we at Safer Medicines Trust believe that there is a new pragmatic way, a new approach to validating new tests by comparing them to animal tests. And how this works is that you select a panel of drugs that have already been marketed, and that they went through all the preclinical testing in animals and in humans, and they looked like they were safe, and they went onto the market, and then they were withdrawn because they were not safe. And so our idea here is to pick those drugs, identify a set of drugs that have been approved, that looked as if they were safe, but they went on to cause serious adverse drug reactions in humans. And for each of those drugs, we select a similar drug which is safe. It doesn't cause those adverse drug reactions. So you have pairs of drugs that act as a control for each other. And the idea is that you test these pairs of drugs with your new human tests, and if that test can distinguish between a toxic drug and a safe drug, then that is what the new test needs to do. That will show that the new test or combination of tests is superior to the animal tests, because the animal tests failed to pick up these toxic drugs. They went into humans and they harmed humans. And so we need to know if the new in vitro tests can pick up these toxicities and can differentiate between a safe drug and a toxic drug. And so this was the study that we've been proposing. Are we all okay? You'll have to wave if we get dropped again. So this study that we proposed is now being conducted by the Evidence-Based Toxicology Collaboration, who are based at Johns Hopkins University, and this is them. Their website is at ebtox.org, and they, as their name suggests, the Evidence-Based Toxicology Collaboration are combining the idea of our study with evidence-based toxicology. And the reason for that is to eliminate bias as far as possible. So they review all of the available evidence, and they do systematic reviews to make sure that the outcomes of the study are robust and meaningful and cannot be accused of bias. And this study has been going on for the past 2 years using the paired compound approach, and it's not quite finished yet, but the first publications are being written, and I will let you know as soon as they're published. But it's very exciting. There are some preliminary results already, and it's looking really exciting and really important, because just on the first pair of compounds, which is troglitazone and rosiglitazone, drugs which were called Vioxx and Avandia, which were both drugs for diabetes. These drugs were marketed for humans. They killed and injured a lot of people, and these harmful effects were not picked up in the animal tests or even in the human clinical trials. And yet they can be picked up, they are picked up by in vitro tests. And so this shows that if pharmaceutical companies would use in vitro tests before going into patients, then we would get much better prediction of safety, and we would spare people in clinical trials and also patients when the drug gets onto the market. We would stop many of the toxic drugs from reaching people in the first place and improve the safety of those medicines. So I'm delighted that we have a new director. I think Javier has explained to you that Dr. Jan Turner is our new director, and she has a huge amount of experience of these new in vitro technologies. And so does Dr. Jerry Kenner, who is our pharmaceutical director. And he gave a talk last week in Cambridge in the UK at the British Toxicology Society annual meeting. And he gave a talk about this study, and he was presenting the initial results of this study. And it's going to go up on our website next week, and I would highly recommend that you listen and watch his presentation, because I won't go into it anymore now because we'd be here all night. But yeah, you will be able to see it on our website next week. Are you hearing me okay?
Yes, everything okay, don't worry.
Okay, lovely. So I'll carry on. And so animal testing has been a mainstay of biomedical research for the past 100 years, and it has been a legal requirement for testing new medicines for the last 50 years or more, ever since the thalidomide tragedy. And so people often worry that if we no longer used animals, that it might be difficult to develop new drugs, as if, you know, there are no other ways to develop new drugs. And so here I want to show you some of the other methods. As we've already mentioned, there are human cells, human tissues. Some of the most valuable human tissues now are human stem cells. And at the beginning, that was very controversial, because using embryonic stem cells is very controversial. But we've moved on a lot since then, and we now have these induced pluripotent stem cells, which are taken from human skin or from human fat cells or from various from cord blood. When from the umbilical cord, you can get a huge amount of stem cells from there. So human tissues, waste tissues from surgery, donated organs and tissues from people who want to do that after they've died. This is the most valuable resource that there is, and a huge amount of discoveries have come from there. And also, since the human genome was sequenced in the year 2000, there's been a phenomenal amount of work done using DNA chips. So there's things like the Human Genome Project, and in the UK we've got the 100,000 Genomes Project, which aims to sequence the genomes of 100,000 people and then compare the DNA between all those different people with and without various diseases. We've got body on a chip devices, which it's the same thing as organ on a chip or human on a chip. There's many different names for the same thing. They're also called microphysiological systems. They're also called microfluidic devices because basically they are tiny bits of human tissue all linked together by a circulating fluid, which is like a blood substitute. And so the result is that it's like it's mimicking the human body on a computer chip, so different organs that are all interconnected. There's clinical research, so that involves patients and giving samples and living patients being studied, and also microdosing, which is, and not just microdosing, but many new imaging technologies, which are absolutely astounding. So we can be imaged non-invasively. You don't have to be cut open or anything. Sorry about that email coming in. But somebody can swallow a drug, a very, very low dose of a drug, and scientists can actually see and monitor how that drug is being metabolized in the body. And so I just want to make the point here, this is a little bit of a diversion, but I just want to make the point that many of our most important medicines were actually developed without animal testing, either because they were developed before animal testing became routine, or for various other reasons. And so aspirin has been in use for thousands of years. People chewed on willow bark, and that's where aspirin comes from. And, Hippocrates was prescribing it in 400 BC. There's an awful lot of serendipity, chance, luck in medicine and in drug discovery. And so antidepressants began because the drugs that were given to TB patients made them feel better. And this was clinical observation. And from those TB drugs, antidepressants were developed. And but there was a lot of self-experimentation in the old days. I wouldn't advocate that now, but it is a fact that, in the old days before anaesthetics, people had terribly painful surgery, and then ether and laughing gas and other anaesthetics were discovered, and people used to have parties and try out the laughing gas and the ether and see how much they could hit each other without it hurting. And in the 1960s, there was an asthmatic doctor who did an awful lot of experimenting on himself. AIDS drugs in the 1990s, AIDS activists were campaigning against animal testing because it was wasting time. They wanted these drugs that had been designed on the computer, tested in human cell culture. They wanted access to the drug. They didn't want to wait 5 years while it all went into animal tests. And leukemia treatments actually came about from nerve gas experiments in the First and Second World Wars, and they have been much more advanced since then, and Gleevec is one of the most successful drugs that we have now against leukemia, and that was designed on computer, and when it was tested in dogs it was so toxic that it was actually thrown away. and it was only rescued and saved by the doctor who had seen its fantastic results in human tissues, and he had desperate patients wanting to try it, and so it was rescued from the dustbin and tested very, very cautiously, of course, in patients, and it is now one of the most successful cancer treatments that there is. And again, cystic fibrosis treatments came about from clinical observation of patients with TB. It was logical and obvious to try those drugs to see if they helped cystic fibrosis, and they did. And as Jerome Groopman said, there's really no other way. If you want to know if a new approach is going to work in humans, you really can't avoid testing it in humans. So going back to the toolbox of methods that we have, methods other than animal testing, systematic reviews are very important. This is what the evidence-based toxicology collaboration is trying to introduce to toxicology across the board, that we get new methods to synthesize all of the evidence, and that will stop people being able to cherry pick evidence that they like and ignore evidence that they don't like. And this should really establish definitively whether medicines are safe or effective by looking at all the evidence that's available. I've already mentioned cell and tissue cultures. We've moved on largely from 2D, two-dimensional models. We've gone on to more sophisticated three-dimensional models, and that includes organoids and organs on chips. Microdosing, I've already mentioned, it's an incredibly sensitive imaging technique. Computer simulation and paired up with bioinformatics and big data, that's very, very important. Mathematical modelling works alongside artificial intelligence, machine learning, systems biology. And what the omics technologies is talking about is things like genomics, proteomics, transcriptomics, metabolomics. There's an awful lot of omics, and so they get lumped together as omic technologies. There's good old-fashioned studies of populations, which is epidemiology. There'll always be a need for clinical trials, carefully, very carefully testing things in volunteers and patients. But one of the most important tools in the toolbox for realising change is the mindset of researchers, and it's also it's the biggest obstacle of all, because a mindset which believes that you have to test on animals to prove safety, this is what we've got to change. We've got to explain and educate about all of these other technologies and change the mindset so that people are willing to try better technologies, because most of these technologies, they're cheaper, they're faster, they do have their own limitations, but in every case the limitations can be overcome. Except for animal testing, the limitations can't be overcome because an animal can never be a human, and so you always have to extrapolate. It's always guesswork. And so what I was trying to say with this picture is that each of these technologies has its own limitations, but by combining them in an intelligent way, not doing all of them every time, but understanding which ones need to be combined in which circumstances, we can get much more accurate, much more reliable results. And so how to overcome the obstacles, particularly the mindset, all of the obstacles to change. We need to change. The need is urgent. It's very important. And it's very, very good news that change is now happening. And one of the main reasons is that we need to work collaboratively. And there are many, many collaborations now, more and more collaborations all the time. There are lots of national initiatives and international initiatives. For example, in America, there's the TOX 21 initiative, which is enormous and very important. In Europe, we have a thing called EU Tox Risk, and this is teams of scientists, teams of academics, teams from industry, regulatory agencies, all working together, because this problem is so vast that nobody's ever going to solve it alone. Everybody is going to have to work together, talk to each other, and this is increasingly happening. And, you know, people have tended to work in their silos, and progress has been so limited because people have not talked to each other enough and not known what's going on in other companies and other universities. And so it's so important that different universities, different companies talk to each other and work together. And the regulatory agencies, the regulators like the FDA, are hugely important because they dictate what tests a company has to do. And the field is changing so fast, these technologies are coming on stream so quickly that it's impossible for the regulators to keep up with them all. So we need to educate regulators about all of the new technologies, and there needs to be a lot of work to find out which are the best approaches, which approaches to use and when. And that means working alongside the regulators and keeping everybody, the public is very interested in this, of course, and politicians. We need to educate politicians, the public, all the different scientists, because a lot of these things are very specialised, and so we need a lot of interdisciplinary collaboration. And this is what's going to really make change happen. And so one thing that we did at Safer Medicines Trust was get together with some other companies and charities, and a couple of years ago we founded a new alliance for human relevant science, and currently we're only in the UK. We do hope that in the future this organisation will become international, but at the moment we're focused on establishing it properly in the UK. And here are some of the founder members. It's much bigger than this now, but we launched the alliance in the House of Commons, in the Houses of Parliament, and we had MPs come along, we had a lot of companies come along, a lot of academic scientists come along. There was huge interest and enthusiasm, and we were so delighted. And here are some of the founder companies. There's Animal Free Research UK, which is another charity like ourselves, but they fund in vitro research. And then some companies which create technologies, in vitro technologies. So here's one of them. This is a company called Kirkstall, and they've invented... Oh, sorry, I skipped on. They've invented a a system called Quasi Vivo, which you can see in the photo. It's quite a small piece of kit, piece of equipment, plastic chambers joined together with tubes with fluid, so you have little samples of human tissue from different organs, for example the pancreas, the liver, piece of fat, all joined together by a circulating blood substitute. CN Bio Innovations is another member of this new alliance, and they produce another sort of liver on a chip type device. They've got it's come to market now. It's called Physiomimics, and it's worth going to all of these websites because they show you videos of these systems in operation, which looks much more impressive than a photo like that. So both of those are examples of organs on chips or human on chip. This is the ultimate aim, is to combine all of these different organs on chips, and so you've got a mini representative human on a chip. And ultimately this can be personalised, so it's not any old human on a chip, it can be you or me on a chip, so that if we have a disease and we want to test a drug to see if it's going to work for us or if it's going to be toxic to us, we can donate little samples of cells, tissues, bits of skin. They can be made into all these little organoids, and the drug can be tested on a mini version of you or me before we get to try it out. And this is from, I hope you can see at the bottom of the screen there, this is from the Wyss Institute at Harvard University. Their website is incredible. I highly recommend you go to the Wyss Institute website and have a look around. It's amazing. And this is an illustrative picture that we were given by a company called Vax Design, who were working on testing vaccines and immune drugs in vitro. And so this is a standard plastic plate with 96 wells that is just standard across the board in every lab, and it's just to demonstrate that you can have little samples in there. You can have a whole representative population, different samples, so you can test different, say 3 different concentrations, 6 different concentrations of a drug in a variety of different people. In vitro, this is a clinical trial in a test tube, a clinical trial in vitro, using real living human tissue, and this is the closest thing to testing in humans with no safety issues at all. So it's absolutely phenomenal what can be done now. It's more impressive than most people realise, and so I'm getting to the conclusions now. And the main conclusion is that what's presenting the paradigm change that we need is simply a lack of confidence in the new methods. It's the regulators are not confident, the regulators are not promoting them as they should be. Scientists who don't work in these areas are not confident. They can't believe how sophisticated these new technologies are. And so we need to be able to demonstrate the superiority of these approaches. That means we need much faster validation, and it needs much more money. So there isn't a lack of science. The science is astonishing, but it needs all the support, the background support that it takes to get a new technology into the mainstream. Is a huge challenge, and governments need to fund this properly. If governments are serious about changing the system and improving safety, then they've got to stump up the funding to enable it to happen. And really importantly, most importantly of all, they've got to set targets. We must have targets, we must have deadlines, we must have incentives to speed up the adoption of these superior methods and to phase out the methods that don't work. And the best example in the world at the moment is the Netherlands. This NCAD at the end, that stands for the Netherlands Centre for Alternatives to Animal or something or other. It's in Dutch, so it doesn't quite make sense to me that way. But they have said that they can phase out the regulatory testing of medicines and other chemicals by 2025. That's what they're aiming to do, is to phase out regulatory toxicity testing in animals by 2025. And it's a wonderful aim. We hope they make it. They need support from every other country, and they have set a precedent which the rest of the world needs to follow. And we will reach a stage in the future, hopefully not in the far distant future, where it will be human on a chip, human organised, human cells and tissues, virtual modelling, artificial intelligence. This will be the norm, and animal models will be left in the past. They will be superfluous to requirements, and they will have been completely superseded. So thank you very much for listening, and if you have any time left, I'm very happy to take any questions.
Thank you very much, Kathy. Yes, I think we are kind of delighted, at least I am. So I guess we have, of course, time for questions.